You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is February 5th, 2024. Our topic today is Abbas Chapter 7, Immune Receptors and Signal Transduction, Part 1, presented by Dr. Nikita Raje. She's an Associate Professor of Internal Medicine, Pediatrics, and is the Chief in the Section of Allergy Immunology at Children's Mercy Hospital.
Hopefully you guys can see my screen. Let's get started. This is one of the most complicated chapters that we'll be talking about from this textbook. So let's get started. We are going to be talking about immune receptors and signaling transduction, Chapter 7 from Abbas. I'm Nikita Raje from Children's Mercy, Kansas City, University of Missouri, Kansas City. Here's my email in case you have questions. And today is February 5, 2024. Here are my disclosures, nothing relevant to the talk today. The outline for this chapter is pretty simple. We'll be talking about TCR signaling, BCR signaling, and cytokine signaling. We'll see how far we get through the chapter. I really want to make sure that you guys ask questions. Ask me to stop if you guys want to kind of catch up or ask me to repeat something. We want to make sure that this is clear. The concepts are clear to you before I just kind of keep moving on and finish the chapter. So if we need to, we'll finish the chapter next time, but we'll see how it goes. Couple of pre-test questions. Which of the following is an example of GPCR? In the T-cell calcium signaling pathway induced by antigen recognition, which enzyme and transcription factor are activated? We'll come back to these. So I wanted to kind of, take a minute to just kind of mention that with this chapter, you learn a lot of new concepts and a lot of new terminology. So kind of, keeping in mind that this is almost like learning a new language again. So in terms of signaling, you want to kind of keep focus on, we are mainly talking about immune signaling, but let's just kind of think about what signaling does. So there are phases of signaling, and the first start of that signaling would need that the receptor binds to its ligand. That binding leads to the signaling, and that can be in terms of a cell signaling. It includes a cytosolic phase and then the nuclear phase. So just want to make sure you see my pointer.
Yep.
Okay. All right. And so let's see. This signaling includes this ligand binding to its receptor, and then the cytosolic phase in the cytoplasm, and then the nuclear phase starts when the transcription factor goes to the nucleus. The exception is in some of the signaling, the end result does not lead to transcription or the nuclear phase, but leads to cellular motility or granule exocytosis, maybe apoptosis, protection from cell death. It can lead to differentiation, it can lead to effector function, it can lead to just commitment to a specific lineage or induction of cell cycle arrest. So a lot of different possibilities of what the signaling leads to. All right, so moving on, let's talk about the first part of that requirement for signaling, which is receptor and ligand binding. So receptors can be on the cell surface, which are cell surface receptors, or in the nucleus called nuclear receptors. Typically, the nuclear receptors have lipid-soluble ligands, versus the cell surface receptors can be cell-bound or soluble ligands. This binding would lead to either cross-linking on the surface of the cell. So lots of, say, for example, B cell receptors coming close to each other, and they're all bound to the ligands, and that causes a cross-linking of all of those receptors. That can lead to activation. Versus in some situations, it would just be a ligand binding to the receptor, leading to a conformational change, and that leads to the signaling or the end effect of what the signaling is meant for. Once that receptor and ligand interaction occurs, we want to think about what the signaling would look like. Now, remember, the signaling has a lot of different parts to what could happen, but typically it involves enzymes that play a big role in signaling. One typical enzyme is somewhere where there is addition of phosphate residue on the side chain of an amino acid in the cytoplasmic portion of the receptor. Again, so addition of a phosphate to the side chain in the cytoplasmic portion of the receptor or some other protein leads to an important step in signaling. Sometimes that phosphate is added to not an amino acid but to a lipid. And so the enzymes that help in addition of that phosphate residue are called kinases. So really important part for immune signaling. Kinases play a big role. But then there are other types of processes that can occur once that receptor binds to the ligand. That could be some other enzymes playing a role, like phosphatases. Instead of adding phosphate, phosphatases remove a phosphate residue. And that also can play an important role. There are other types of post-translational modification of the receptor, like acetylation or methylation or ubiquination, that also play a role in signaling. Now, there are different types of signaling receptors, right? We know that there are what are known as non-receptor kinase, tyrosine kinase-based receptor. What does that mean? So, as the name suggests, the tyrosine kinase. So tyrosine kinase is a kinase, which means it adds a phosphate molecule to tyrosine. So kinases add phosphate molecule to any amino acid like serine or threonine or tyrosine, and the tyrosine kinases are typically really important in immune signaling. So we'll go back to tyrosine kinases quite a bit in this chapter. So here we are talking about a tyrosine kinase that is not on the receptor itself. So here is a receptor, right? And the tyrosine kinase is the small structure here, which is not a part of the receptor itself. Those are called non-receptor tyrosine kinase based receptors. Then there are tyrosine kinase receptors that have the tyrosine kinase as a part of the receptor structure itself. There are nuclear receptors, which means the nuclear hormone typically goes and it's an intracellular receptor where the nuclear hormone, for example, will go in and attach to that receptor, and then that whole structure will translocate to the nucleus where it will help with the transcription of various target genes. So those are called nuclear receptors. Then there are GPCRs where they span the membrane 7 times and they help with activation of G protein. So that G protein plays an important role to help in activation of other enzymes like adenylate cyclase and phospholipase C. Then there are receptors that use proteolytic cleavage, which means that the receptor is one structure, but it has 2 parts to it. When it attaches, when that receptor binds to its ligand, in purple here, that ligand leads to the cleavage of the 2 parts of the receptor itself. So there is like a break in that receptor. The part that's broken and cytoplasmic part that's broken into 2, basically that second part moves on to the nucleus to help with signaling. And so examples of some of these receptors, so non-tyrosine kinase receptors are pretty much all immune receptors. So B cell, T cell, cytokine receptors, integrin receptors. the tyrosine kinase based receptors, which means the tyrosine kinase is part of the receptor itself. Some of the examples are c-KIT receptor, or epidermal growth factor receptor, insulin receptor. Nuclear receptors are steroid receptors, glucocorticoid receptors, or vitamin D receptors, GPCR, histamine, prostaglandins, complement receptors. So there are several of those examples. And then Notch receptor is a type of proteolytic cleavage type of receptor, which is important in T cell development. Sorry, going back here. Yeah, we covered everything. All right. So apart from the receptor itself, we want to talk about the signaling proteins. So, so when the receptor binds to its ligand, which we just talked about, the next step is using some of these signaling proteins and enzymes and adapter proteins. So when we talk about the enzymes, we talked about kinases and tyrosine kinases, and the tyrosine kinases come in different families. They are called Circ family or Sic family, Tech family. And what does this mean? So we have heard about modular furniture, right? Small pieces that can be brought together to form a bigger shape. And or you can think of Legos. Small pieces of Lego, you can bring them together to make a big structure, lots of different pieces, lots of different shapes and sizes of pieces that fit into one another to form a big structure that actually has meaning to it, right? So think of these tyrosine kinases as that. These tyrosine kinases are different types: Circ family, Sic family, Tech family. So now let's look at the kinase part of it. This blue part here has the kinase enzyme, but each family has other structure to it that plays a role in what it binds to. So those parts are modular, which means each piece of it can be bound to another piece to give it a structure. So one of those pieces is called SH2 domain, SH3 domain, or pH domain. So SH2 domain, for example, binds to a phosphotyrosine, which means a tyrosine that's already phosphorylated. That's what the SH2 is going to bind to. SH3 is going to bind to a proline-rich peptide. And then pH domain binds to inositol phospholipids, right? And then there are some of those unique domains that might be different in different kinases in each family. So if you look at the Circ family kinase, it has one SH2, one SH3 domain, whereas a Sic family has two SH2 domains, and that's important in what we are going to talk about next. Similarly, Tech family, remember that those pH domains help with binding to phospholipids. So these are the tyrosine kinases, or the enzymes, right? Now we talk about adapter proteins. So we talked about how those enzymes or signaling molecules are modular. So they come together, different structures come together to form a bigger, a Lego, for example, a Lego set that's coming together to form, give structure as well as function to that enzyme. Apart from the enzymes, there are adapter proteins. These adapter proteins do not have any enzymatic activity to it, but they just act as hubs or docking sites. So we all use these hubs, different types, to connect different electronic devices so that they are connected to each other. So these adapter proteins are like hubs that connect different enzymes or different proteins that come together so that the signaling cascade can proceed. So these adapter proteins are docking sites that physically link different enzymes and promote the assembly of signaling molecules. These could be on the membrane, or they could just be in the cytoplasm. Some of the examples are something called LAT, SLP76, GADs. So different ones, different adapter molecules. They do not have any enzymatic activity, again, as I mentioned, but they act as docking stations, so the enzymes and other proteins come together so that that structure continues to progress and then signaling continues. These docking sites or adapter proteins also have similar domains that we talked about: SH2 domain, SH3 domain, proline-rich peptides regions, and then tyrosine residues. And so all of these structures decide what they will bind to. All right. Now, let's talk about the receptor families. So we want to kind of start talking about immune receptors. We talked about receptor and ligand binding, signaling molecules that have enzymatic activity, and adapter proteins. That was just the basic structure and basic information about the terminology that we are going to be talking about as we talk about these immune receptors. So we know that the immune receptors that we are interested in are typically the T cell receptor or the B cell receptor, some of the FC receptors, or some of the inhibitory receptors that are part of the immune receptor family. Now, you know this structure very well from our discussions in the past chapters about immunoglobulin. That's that particular beta-pleated sheet structure that is typical of immunoglobulin domain. And not just the immunoglobulins, but a lot of molecules in the in immunology have this particular immunoglobulin domain as a part of their structure. They all belong to immunoglobulin superfamily region, right? Or they belong to that superfamily, including the BCRs, the TCRs, FC receptors, as they are all shown here. Now, if you look at these receptors, let's look at PCR. There's this whole structure outside the cell. This is the cell membrane. But if you look at the cytoplasmic portion, it's very small. Similarly, TCR, it has this big ligand binding portion, but then the cytoplasmic tail is pretty small, similarly for the other receptors. So the cytoplasmic tail that would typically help with signaling is so small that typically you need another signaling protein that is attached to the receptor itself that has a longer cytoplasmic tail that helps in signaling. That's called transmembrane signaling protein. It's different for different receptors. Like for PCR, it's Ig beta and Ig alpha. For TCR, CD3 and zeta chains, and so on and so forth. All these signaling molecules at their cytoplasmic tail have a tyrosine-containing motif. These tyrosine-containing motifs can be two different types. The one here that you see on all these receptors, but you look at the last two, which have the purple one, which is called ITIM. ITAM is the one that you find in BCR, TCR, and some of these receptors that stands for immune receptor tyrosine-based activation motif. So these are activating receptors or signaling molecules, versus ITIMs are immunoreceptor tyrosine-based inhibitory motifs. So ITAMs are activating, ITIMs are inhibitory. These ITAMs and ITIMs, or overall the signaling proteins that are next to the receptor, are in close proximity to the kinases. Why is that? The kinases, the Circ family kinases, are the ones that can actually add a phosphate molecule to the tyrosines. Those tyrosines are present on these ITAMs. All right, so as you see here in the structure of ITAMs or ITIMs, right, there's a very particular structure that has tyrosine in them. They also have some other amino acids, as you mentioned them, but the ITAMs and ITIMs have a very particular structure to them. All right. So think of the ITAMs or ITIMs to be the wick, and then they are almost like the ignition points, and the ignition to them is provided by these Circ family kinases. And that helps the enzymatic activity in that kinase, adds the phosphate molecule and ignites that ITAM so that the signaling can proceed. With me, everyone? Any questions? Okay.
We're with you.
Go ahead.
I said we're with you for now.
Okay, sounds good. So Say the receptor binds to its ligand. And I showed you all these receptors that have several items for TCR, multiple for BCR, multiple for these FC receptors, some of these FC receptors. So you can see that it's not just an on-off switch, right? When that item, it gets that ignition, it's not just that it's going to be activated. The T cell receptor is gives the activating signal. There are so many items that the receptor signal strength can be different in different interactions. And so it's almost like the sliding switch rather than the on-off switch, where number of items that are used decide how strong the signal is for T cells or B cell signaling. Also, the signal strength can decide whether that cell is going to be activated and it's going to progress through signaling, or during maturation, the binding of the same kind of items can lead to either survival of the T cells, or if it strongly binds to a self-antigen, that TCR or BCR will lead to binding or ignition of those items and lead to the cell death of those cells, because they were able to bind to and send the signals through the TCR and BCR so strongly, and they are they can bind to the self-antigen that can lead to autoimmunity. So the immune system basically decides that the T cells and B cells need to undergo apoptosis. So the effect of the receptor ligand interaction and the effect of the signaling strength can lead to different kinds of end results. Also, this apart from the items binding to the coming in play in the receptor ligand activation is further changed based on the binding of the co-receptors, the function of the co-receptors. So in case of T cells, CD4 or CD8, in case of B cells, CD21. The inhibitory receptors of T cells and B cells can also play a role, such as the CTLA4 or PD1 in case of T cells, Fc gamma R2b in case of B cells. And then again, it's the second signal that's provided by CD28 binding to either CD80 or CD86 can further modulate the signaling that's provided by lymphocytes. All right, now we talk about T cells first, and then we'll talk about B cell signaling. So the signaling starts with the receptor. So let's talk about T cell receptor. T cell receptor is two types, alpha beta or gamma delta. 95% of our cells are alpha beta, 5% gamma delta on an average. Now, we know that the alpha beta receptor itself has it has the alpha chain and the beta chain, as I mentioned, but it has a very small cytoplasmic tail. So it needs the bells and whistles that come along with the signaling molecules. And in case of TCR, the signaling molecules are called CD3 and zeta chains. CD3 is a heterodimer, two different heterodimers, epsilon and gamma and epsilon and delta. So two heterodimers and a homodimer of two zeta chains. All of them together are associated with the TCR to help with the signaling. Now remember, as you look at the cytoplasmic tails of these signaling molecules, you'll see each of the CD3 chain will have one item, and the zeta chains actually have three items on the cytoplasmic tail. Now remember, items stand for activation motif, which means when these are phosphorylated by a kinase, there will be activating signals that will go through this TCR. The table talks about how there is a difference between immunoglobulin receptor and the T cell receptor, and how they are different. But in general, there are these the binding portion of the TCR, which is alpha and beta, those domains have one variable domain and one constant domain, and the variable domains are the ones that can bind to various types of antigens. So they have a lot of variability in their structure, and that variability is the highest in the CDRs, just like the immunoglobulin receptors. So here's again the structure of TCR. The variable region is called the V beta or V alpha for beta and alpha chains. And then they bind to the peptide, right? This TCR binds to the antigen presenting cell along with its MHC molecule that shows the peptide or presents the peptide to that TCR. So the TCR portion, alpha beta portion with its variable regions is the one that binds to that peptide. Here is class 1 MHC with its beta 2 microglobulin chain. Here's your TCR and different kind of view from the top showing the variable and the constant regions. Co-receptors, CD4, CD8 for T cells. Here is the CD4 structure, all these immunoglobulin domain structures, and then CD8 has 2 chains, alpha and beta, each with 1 immunoglobulin domain. The cytoplasmic tails bind to the Circ family kinase. So just kind of looking at this here is an antigen presenting cell with its MHC. This is a CD4 cell. So CD4 has, and the MHC class 2 is presenting the antigen. Now, if you look at this T cell membrane has the TCR itself, right? So here's our TCR, and it is interacting with its signaling molecules, CD3 and zeta chains. Close to the TCR is the co-receptor. So close to the receptor is our co-receptor. In this case, it's a CD4 cell. So it's a CD4 because it's a CD4 cell. So CD4 on the surface, it's going to be in close proximity to LCK, which is a Circ family kinase. What does a kinase do? It adds phosphate to some of these items, right? Circ family tyrosine kinase, which means it provides a phosphate to one of the tyrosines that are present in these items. So the LIC or LCK is one of the Circ family kinase that is in close interaction with its co-receptor, T cell's co-receptor. All right, so all this interaction that we saw, in between these two cells occurs in this small space between the two cells. And so we want to pay attention to how these two cells are kept close to each other. So the close interaction between the T cell and antigen presenting cell occurs in this small space that's called supramolecular activation cluster. It's called a cluster and the cluster itself has 2 parts to it, central and peripheral, C-SMAC and P-SMAC. P-SMAC or peripheral SMAC is where the adhesion molecules keep the 2 molecules close to each other. So the integrins binding to integrin ligands and keeping those 2 cells close to each other. And then the central SMAC is where all our interest is, right? The TCR binding to the peptide and MHC class 2, co-receptor, second signal or co-stimulators, all coming in interaction with each other between the 2 cells. And so all these molecules are present on these microdomains that can actually move close to each other so that that interaction can occur. Now, if it was a CD8 cell instead of CD4 cells, there could be delivery of granules between these 2 cells, versus in the case of both CD4 and CD8 cells, there could be exchange of cytokines or just kind of release of cytokines between these 2 cells. All right, so for the P-SMAC and C-SMAC, just kind of thinking of what the various molecules are and they come in interaction with which kind of molecules, just kind of looking at those combinations here that I just mentioned. All right, with that, I'm going to go ahead and start with talking about the signaling itself. stop me anywhere that you need to. But we talked about the adapter molecules, signaling molecules, receptors. We talked about T cell receptor, and the T cell receptor coming in contact with its antigen, and then all the signaling molecules that play a role in that T cell signaling. So when we talk about signaling, I want to kind of, we talked about all this stuff that would lay a foundation to what we are going to talk next. But now I want to reverse that discussion and think about the end result. What's the purpose of signaling? What are we trying to do here? What's a T cell doing when that receptor binds to its interaction with its ligand? What does that interaction do? The purpose is that the T cell gets activated. When that T cell gets activated, the whole purpose of the T cell being activated is that the T cell undergoes clonal expansion. The T cells proliferate and then differentiate. They differentiate into a effector cell, like a cytotoxic cell, or a helper subset. And that cell would have effector function of either releasing cytokines or helping with cytotoxicity. Along with that, it's important that that activated T cells continues to survive for a longer time to help with that function. So all these things have to occur once that receptor binds to its ligand. And all of these changes occur because of the signaling. So why the signaling is important? Because that leads to the end result. So I want to keep the end result in focus, because that's how we are going to decide what happens with that signaling. So receptor binds to the ligand, and there is a geometric shape creation either by cross-linking or conformational change. That leads to signaling pathways. So various signaling molecules and enzymes and adapter molecules bind to each other, and that leads to activation of transcription factors. The transcription factors then translocate to the nucleus, and they bind to the promoter region of particular genes. That affects the gene transcription. And so transcription leads to formation of mRNA that code for proteins needed for these functions that we just talked about. So for example, T cell signaling would lead to the end result of production or transcription of IL-2 gene that leads to production of IL-2. What does IL-2 do? IL-2 helps with proliferation of T cells. IL-2 helps with differentiation and function of T cells and helps with survival of T cells. So keeping that just a very narrow focus as we talk about signaling helps us to understand why the signaling is important and what's the signaling trying to lead to. So the signaling is trying to lead to various genes being transcribed, and the major part that we are trying to see is how it leads to activation of transcription factors. The transcription factors can be helping with transcription of various genes, but in T cell signaling, of course, there are a lot of genes that are affected. But for an example, just to keep it in simple, we'll think about like IL-2 gene being transcribed. So the transcription factors that are important that need to be activated so that this purpose is solved, right, are AP1, NF-kappa B, and NFAT. So our goal for signaling in T cells is a receptor binding to its ligand, leading to the activation of transcription factors AP1, NF-kappa B, NFAT. The end result of these transcription factors would be them going to the nucleus, helping with transcription of various genes that lead to the survival, proliferation, and function of T cells. All right. So keeping that in mind, let's talk about signaling. So we start with the same process again. Receptor binding to the MHC complex on the APC bound to its peptide. So the alpha beta TCR binding to the peptide is the first signal. That signal needs help from the co-receptors. So the TCR, here is our TCR, binding to the peptide and MHC class 2 on the APC. Once that happens, it is co-clustering with the CD4 or CD8. So if this is MHC class 1 versus MHC class 2, it's binding to the co-receptor, and the co-receptor has LCK bound to its cytoplasmic tail. LCK or FIN, another type of Circ family kinase, are kinases, tyrosine kinases, that lead to phosphorylation of ITAMs on the CD3 or signaling molecules of the TCR. Once that tyrosine is phosphorylated, it leads to binding of zeta-associated protein or ZAP70 that comes close to these phosphate molecules. So phosphorylated tyrosines. So ZAP70 comes and attaches to those phosphorylated ITAMs, and then that ZAP70 acts as a docking station for various molecules like LAT. LAT, remember, is an adapter protein. Now the item on the LAT will get phosphorylated, and that LAT helps as a docking station for various enzymes to come and bind to it. Some of them being PLC gamma 1 or MAP kinase proteins. So keeping this in mind, this will lead to activation of MAP kinase pathway and PLC gamma pathway, or phosphorylation activation of PLC gamma pathway. The PLC gamma pathway then leads to two different paths, the DAG and IP3. PLC gamma leads to phosphorylation and activation of this PIP3. So we'll take a look at that in a minute here. PIP2 to PIP3, that leads to DAG and IP3, and that leads to three different pathways: NF-kappa B, NFAT, and AP1. MAP kinase is the one that will lead to activation of AP1. So these are the three transcription factors that will be activated. So this is the initial common pathway of signaling. So let's look at it again. Receptor binding to the ligand. Co-receptor is in interaction with that MHC complex. There is a second signal that's provided, that all of that leading to activation. That leads to LCK, lick, phosphorylating ITAMs on the signaling molecules of TCR, right? CD3 and zeta chains. That phosphorylated tyrosine act as a docking station for ZAP70, and ZAP70 again coming in contact with becoming a docking station for LAT. LAT becomes a docking station for PLC gamma as gamma 1 and MAP kinase proteins. Apart from those three, one of the important change that happens with that receptor ligand binding is cell survival pathway. So the cell survival is separate from those three transcription factors, and so it's important that this T cell that got activated survives. How does that happen? PI3 kinase. We know PI3 kinase pretty well, right? PI3 kinase defects lead to immunodeficiency called APDS. PI3 kinase helps in phosphorylating PIP2 to PIP3 on the cell membrane. So PIP3 then leads to activation of something called PDK1. That leads to activation of AKT. And AKT leads to inactivation of apoptosis factor called BCL2. So because of that, the cell survives longer because the apoptosis factors are inactivated. So PI3 kinase is an important cell survival pathway that gets activated. The signaling leads to the survival of the T cells through PI3 kinase. All right. Now we talk about the rest of the pathway that is separated after the LAT or LAT that binds to various or leads to activation of various pathways. So that pathway was TCR binding to the receptors, its ligand, and that leads to LCK phosphorylating the ITAMs. Then the ZAP70 comes in contact with it, and then the LAT. Once that LAT is phosphorylated, there are two types of proteins that come to dock onto that LAT called GRAB2, and GRAB2 then interacts with SOS. GRAP2-SOS leads to interaction so that the RAS GDP is phosphorylated to RAS GTP, right? Diphosphate to triphosphate. And that RAS GTP goes to the nucleus and leads to the activation of RAF, MEC1, and then the most important one being ERK. So ERK is activated ERK binds to ELK, and activated ELK leads to transcription of something called CFOS. Now remember, this MAP kinase pathway is going to help us get one transcription factor activated called AP1. AP1 has two parts to it, CFOS and CJUN. And so this whole pathway of GRAP2-SOS phosphorylating RAS GDP to RAS GTP ends up in transcription of one part of AP1 called CFOS. And that pathway from RAS GTP is that MAP kinase cascade that leads to formation of ERK, and that activated ERK then basically leads to the activation of ELK. Activated ELK leads to transcription of CFOS. On the other hand, the LCK, by this whole process where LAT is docked, LAT helps in docking station of not just this GRAP2-SOS, but a very similar process where instead of GRAP2-SOS, it leads to activation of VAV. VAV leads to formation of RAC GTP, similar to RAS GTP. RAC GTP helps in recruiting JUNK or SAP kinase, and activated JUNK phosphorylates CJUN. CFOS and CJUN together form the activated AP1, which is one of the transcription factors needed. That AP1 is now going to go to the nucleus where it's going to bind to a promoter region and help in transcription of genes that are needed for the survival, proliferation, and function of T cells. All right, so this is one of the important pathways for the signaling pathways of T cells that helps with the function of the T cells, right? All right, now we move on to other pathway. So again, the same pathway that we talked about. Antigen binding to the receptor coming in contact with the co-receptor, and then there has to be a second signal, that is the co-stimulator, not shown here, and all of that leads to LCK at the cytoplasmic tail of co-receptor. Helping, what is LCK again? Cert family kinase. It phosphorylates tyrosine. So tyrosine in the ITAMs is phosphorylated. That leads to so the one in the zeta chain leads to activation of zeta associated protein, or ZAP. ZAP70, once it can bind to those phosphorylated tyrosines, can help with engage LAT, and that LAT can lead to becoming a docking station for various things. We talked about the MAP kinase, and it also becomes a docking station for PLC gamma 1. Here is PLC gamma 1. Once it's activated, it also, along with PLC gamma 1, it also interacts, LAT also interacts with ITK. Think of ITK similar to BTK in B cells. So ITK gets engaged, and that helps with PLC gamma 1 activation, and PLC gamma 1 then leads to formation of two parts, IP3 and DAG. So dialysylglycerol and inositol triphosphate. These two molecules then lead to two different pathways. So let's talk about IP3. So there's IP3, PLC gamma leads to formation of IP3, and IP3 uses the calcium pathway. So what does it do? It leads to the activation of STIM1 that releases the intracellular calcium stores. And so this is endoplasmic reticulum. These are the intracellular calcium stores that release the calcium. Once that PLC gamma acts on these stores, the calcium is released from the intracellular stores into the cytoplasm. Because of that, the STIM1 is a sensor of calcium intracell in those stores, and it can sense that there is low calcium. So that STIM1 is going to go to the cell membrane and bind to a channel called crack channel. There is a protein called ORI in that channel that helps to open up that channel so that the calcium can come into the cytoplasm from extracellular space. So that calcium that comes into the cytoplasm from the intracellular stores and the extracellular space, all of that calcium leads to increase in cytoplasmic calcium. Because of that, there is interaction of calcium with something called calmodulin, and the calmodulin and calcium together help in activation of calcineurin and NFAT. NFAT is the one that leads to transcription of particular genes. So NFAT is going to be acting as a transcription factor. Let's talk about the second pathway, which is the IP3. We will, we talked about IP3, we'll talk about the DAG pathway. I guess the DAG pathway is, I guess it will be a minute before we talk about it. Okay, all right, so we are still talking about the NFAT pathway. So let's talk about that rest of the pathway in that, and the various molecules in that pathway. So NFAT, calcium and calmodulin dependent calcineurin phosphatase activates NFAT, and then that translocates to the nucleus to give rise to various gene transcription, various molecules that can be released like IL-2, IL-4, TNF. Trying to see if I can show you here. Okay, so remember the calcium in the cytoplasm was increased because of IP3 and STIM1. That calcium will bind to calmodulin, and the calcium calmodulin dependent calcineurin phosphatase will help in activating the NFAT. So NFAT is going to be, it's a phosphatase, so it removes that phosphate. Now, in this case, the removal of phosphate leads to activation. And so active NFAT will go to the nucleus where it will help with transcription. Remember, there are some drugs that are used so that they are called calcineurin inhibitors, and those calcineurin inhibitors are used to prevent this cycle signaling cascade so that the IL-2, TNF, IL-4 are not released, so that the T cells are suppressed. So these are used as immunosuppressants such as cyclosporin or tacrolimus, and these drugs are calcineurin inhibitors that bind to particular binding proteins like cyclophilin in case of cyclosporin or FKB binding protein for tacrolimus, so that they can help with immunosuppression. All right, just kind of moving through, wanted to talk about this protein kinase C pathway, which leads to, so the IP3 DAGs, right? So remember the LAT, this part says the same, LCK, ZAP70 LAT, that interacts with ITK and PLC gamma 1. PLC gamma 1 leads to IP3 as well as DAG, and that DAG leads to activation of protein kinase C. Protein kinase C theta is in T cells, PKC beta is in B cells. That PKC, that cycle or signaling cascade leads to the activation of transcription factor NF-kappa B that leads to, again, transcription of various genes. So we'll come back to that. NF-kappa B pathway is also important for other other signaling cascades, not just T and B cells, but toll-like receptors, other innate pathways, cytokine signaling. So that pathway we'll talk about in a minute. All right, so we talked about the signaling in T cells. Remember, this T cell signaling needs a lot of regulation, and that regulation is really important because T cells can help with immune response, but it can also cause inflammation, so it needs to be regulated. There are a lot of non-coding RNAs. Apart from the mRNAs that are transcribed, some of the signaling can also help with some non-coding RNA. Those could be long non-coding RNA, small non-coding RNAs, or microRNAs. And the microRNAs help with with the regulation of T cell signaling. Some of the information here about microRNAs is mentioned here. The microRNAs are transcribed from the gene as primary transcript by an enzyme called DROSHA, and then that is some of the introns are removed to make pre-miRNA, pre-microRNA, by an enzyme called dicer. The mRNA, right? mRNA is the coding RNA that interacts with the microRNA, which is the non-coding RNA, and some proteins called organoid proteins. All of those together, when the microRNA binds to the mRNA in this complex, it actually silences the transcription from the mRNA, or silences the mRNA so they're not translated, sorry. So that complex is called RNA-induced silencing complex, or RISC, R-I-S-C. So that's how these microRNAs can regulate T cell signaling. All right, apart from the non-coding RNAs, there are other proteins that help with regulating T cell signaling. We know that the kinases add phosphate to some of these, some of the tyrosine kinases add phosphates to the various amino acids and lead to activation signals. So there are phosphatases that help in taking away the phosphate molecule so that they can stop the signaling. And some of these are called SHP1, SHP2. They interact with tyrosine kinases. They are phosphatases that kind of counteract the tyrosine kinases. There's also SHP. SHP is the one that counteracts PI3 kinase. So it will lead to lack of survival of the cells. And then there is a role of another phosphate, which is called CD45. CD45 has a role in actually activation. So we'll look at that part in a minute here. There is also something called a switch signal, CD2 or SLAM co-stimulator. CD2 family has a protein called SLAM that the cytoplasmic tail of that has something called not an ITAM or ITIM, but an ITSM, which is an immunoreceptor tyrosine-based switch motif, which means it can switch from an activation to an inhibition signal. So it can do both. There are that SLAM signal, there is a protein in that SLAM pathway called SAP, SLAM-associated protein. Defect in SAP caused by mutation or variant in gene called SH2DIA leads to X-linked lymphoproliferative syndrome. One type of XLP1 that is caused by SAP defect. There's another type of switch motif in 2B4 that also acts as a switch motif that can immunomodulate this signaling in T cells. All right. So I told you how CD45 actually helps with activation. So what is that? So remember, when the T receptor, TCR, binds to its antigen, the co-receptor CD4, for example, or CD8 has LCK bound to its cytoplasmic tail. At rest, when that receptor is not bound to its ligand, that LCK is inactivated. So it is bound to the TCR's cytoplasmic tail in a conformation where it's inactive. When there is a CD45 that comes in that makes it possible. So CD45 is a phosphatase, and what it does is it takes away the phosphate from the SH2 domain that is bound to the LCK. When that the SH2 domain is dephosphorylated because of the CD45, which is a phosphatase, the LCK changes its conformation so that it's available to phosphorylate the ITAMs on zeta chains. So even though CD45 is a phosphatase, it helps in activation. versus CSK is the reverse. When that CSK comes along, it will add phosphate molecule. It's a kinase. It will add phosphate to the SH2 domain so that the LCK gets wound up again and is in inactive stage. So remember, CD45 is a phosphatase but helps in activation of T cells. There are metabolic changes that occur in T cell activation. In the resting phase, the T cells use mostly oxidative phosphorylation, so may produce a lot of energy, a lot of ATP molecules, versus when they are starting to proliferate, they are using up a lot of energy. They use aerobic glycolysis. All right, I think